got the slide deck right on time. So our next speaker is Tiffany Hsu.
Yep.
If I pronounced that right. From the Broad Institute in Harvard.
Hi, thanks for the introduction. This is work from Curtis Huttenhauer's lab, and so I'll be talking today about detecting lateral gene transfer from assembled metagenomes. So first, a quick overview on what lateral gene transfer is. So microbes can not only get DNA from their parents through duplication, but they can also get genes from their friends. So this happens mainly in three ways. In this case, I have the purple microbe getting genes from the orange microbe, and sometimes this ends up with the genes from the other organism incorporating into the genome, as I've drawn here. Other times, those other genomes come in as extra chromosomal elements or as plasmids, but the main point is that by getting new genes from other microbes, you're gaining new functions. And this has been particularly important in, say, antibiotic resistance. This is from a WHO report showing E. coli gaining resistance to many different antibiotics, and some of these came up primarily through lateral gene transfer. So if one microbe can have such a change in phenotype, since we're at the microbiome cozy, a question we can ask is, how much does lateral gene transfer affect a microbial community? And to answer this question, we first have to know if we can detect lateral gene transfer in these communities. And most communities, or at least in the Human Microbiome Project, have been sequenced using short-read sequencing. When you assemble those into contigs, they're relatively short, while a lot of the tools and techniques for searching for lateral gene transfer are for whole genomes. So if we can start to detect lateral gene transfer in these shorter assemblies, we can start to look for lateral gene transfer, say in a community like the human microbiome, and ask how often is lateral gene transfer occurring, which microbes are exchanging genes, and what genes are they exchanging. So to do this, we generated a method, and it's called Waffle, which stands for Workflow to Annotate Assemblies and Find Lateral Gene Transfer Events. And the way it works is, so I've drawn the genome from before on here, and if you shotgun sequence this and you get contigs, these contigs are fragments of that genome. And you don't know which tags are on there or what genes, so I'm taking away the colors and the arrows. But what we do first is a nucleotide alignment against a species pangenome database. So in the database on the right here, each box represents a species. Within each box is the pangenome for that species. So we do a nucleotide alignment of contigs against the genes themselves. And from that, you can either call genes using those hits, the start and end sites, or you can supply your own gene calls. But once you have your genes and your hits, you can start to assign taxonomy and function to each gene along the contig. And from there, you can decide if your contig, if all the genes come from the same organism, we call it as no lateral gene transfer. But if the genes from a single contig belong to two organisms, we then classify it as lateral gene transfer. So to validate our tool, what we did was to generate synthetic contigs. So here is an example of that. We'd have a 3-gene contig with two organisms, A and B. A and B can differ at these are the same kingdom, so the last common ancestor is the kingdom, but they're interphylum transfers all the way down to where they share the same genus, but they're interspecies lateral gene transfer contigs. And we always know that our reference database is missing things, so what we did was a holdout. So you had your contig to gene alignment. What we would do is withhold up to 20% of those alignments to simulate what would happen if you're missing a gene from the database. So what we can then look for is to see how well does Waffle call lateral gene transfer when we created a contig that supposedly has it versus, and how many false positives does it get. And it performs pretty well down to intergenus transfers. And even when we create longer contigs, it still does pretty well. So with our validation, we then moved on to run Waffle on the expanded Human Microbiome Project 1 dataset. So the first thing we did there was to look at the different sites. So right now I'm just showing the stool, but the upper graph is to show the lateral gene transfer frequency. So each point is a metagenome, like one person's sample. And on the top, we have the number of lateral gene transfer contigs divided by per 1,000 genes. While on the bottom, we have more the assembly size, but is total number of genes in that metagenome. And we can start comparing this to other sites. So here in the blue are the oral sites. And we can see that the plaque and the tongue dorsum also have pretty high numbers of transfer. Whereas when you start looking at the skin and the vaginal sites, the amounts are lower. And some people have pointed out, like this kind of does correspond to how many genes are in your assemblies. But that also reflects the diversity of the community. So the stool and oral communities are pretty diverse and have a lot of different microbes that can do transfer, whereas that's not so much the case for like a vaginal site. But from here, we can start to zoom in on which microbes are transferring. So I'm just going to focus on supragingival plaque from here. And here I'm showing the top 10 genera from the supragingival plaque. And so the order of the rows and across the bottom, the columns are the same. But basically here, yellow means more transfer, while purple means less. So you can see some bugs like actinomyces can be kind of picky. They like to transfer with, say, Corynebacterium rothia, but not so much with the last three. Whereas strep, it's kind of the same color throughout. They're not as picky about who they transfer with. But you can start looking at pair characteristics and seeing if there's general trends for lateral gene transfer. And one of those ones we looked at was phylogenetic distance. So if you're short phylogenetic distance, you're closer on the tree. But if you're far apart, you have a larger distance to traverse along the tree. But we can start to see for each pair, each dot here is a pair of microbes, whether they have more transfer or less. And as we kind of expect, things that are closely related are transferring more than those that are farther apart. For pairs, we can also look at their abundances. So by co-abundance, I mean the abundance of each bug multiplied. And in this case, we see that bugs that are also very abundant also just tend to transfer more than bugs that are pretty rare. And I think that's interesting because it's kind of like if there's more chance to run into each other and just transfer, like it'll just inevitably happen. And some of it might stick based on phylogenetic distance, while others might not. So lastly, we went to look at function. And in red and yellow here means these functions were enriched in contigs with lateral gene transfer, while in blue means they were depleted. Most of these in the green and blue are DNA binding proteins or mobile elements, so like transposases and resolvases. What's interesting is these TOMB and outer membrane beta barrel proteins are up, but some of these membrane and transport proteins are down. And these indicate some antibiotic resistance proteins, but they also seem to be down, even though we know they are often transferred as well. And maybe this is because it's a healthy cohort they're not being selected for. And to tie it all together, I'm going to show one contig that has genes from this clan. So what Waffle will give you is, in this case, we have a single contig that's about 10,000 base pairs. The y-axis is sort of the alignment score. And each of these arrows is an alignment to the contig. And the blue is colored for Haemophilus, the genus Haemophilus, while the red is hits from the genus Aggregatibacter. And so this is an example of what you would see in Waffle and call as a lateral gene transfer contig. And we annotate functions wherever there's a UNIRF90 available. So in summary, we generated a method that detects lateral gene transfer from short-read metagenomes. We evaluated it and then used it to catalog lateral gene transfer in the Human Microbiome Project. And we see more transfer in the stool and oral sites. Between pairs, phylogenetic distance and abundance matters. And for function, we see enrichment for mobile elements and less so for some of the antibiotic resistance proteins. So with that, I want to thank the Huttenhauer Lab, especially Curtis and Eric, the Dalhousie Dream Team here, and Chongwei from the Broad, and as well as ISCB for giving me a travel fellowship to come here. So if you have any questions, feel free to email me, or we have a Google group on the website.
Time for perhaps one question while we change over speakers. Can the next speaker please come up?
So I would be curious if I was intrigued with your antibiotic resistant gene data. So I was curious if different mechanisms of resistance showed different types of stability. And as you probably know, these genes are ancient evolutionarily. I mean, they go way back into the start of evolution. And they have probably many functions that are important that were relevant before antibiotics.
Mm-hmm.
So the question is, I don't understand the data. It's just intriguing. So if you had any further thoughts on it.
Yeah, part of it is we don't really know directionality. So for our contigs, we know the contig has transferred, but we don't know which genes in that contig were actually transferred for the function.
Are they from the plasmid or from the genomes? Or?
That's also hard to say. As long as it's a fragment of DNA with annotations from 2 organisms, we're sort of just calling it as transfer. But with the contigs, as some of them as short as they are, it's hard to tell if they're from the genome or, say, like a plasmid.
Okay. Thank you. We'll have our next speaker now.